capillaries. All right. Arteries always carry blood away from the heart. Veins always carry blood back to the heart. And capillaries are very thin-walled vessels, which are too numerous really to name, and they're there for exchange. Okay, their walls are so thin and their diameters are so narrow, right, that red blood cells have to go through like a stack of coins, sliding through one column at a time. And as they do, they rub against the walls of the capillaries, and that's actually where they have most of their damage take place. But at the same time that's going on, we are dropping off oxygen at the tissues, and at the other end of the capillary bed, we're picking up carbon dioxide, all right? And it's the oxygen-poor, CO2-rich blood that's eventually going to return to the right side of the heart, be pumped back to the lungs, be reoxygenated, and then from there be pumped back to the left side of the heart, delivered to the left side of the heart, and then eventually into the left ventricle, where we'll pump it into the aorta, and that'll take it out to the body again. Okay? The arteries are a high-pressure system. Okay? The blood that they carry exerts more force against the wall of the vessel, so the wall of the vessel is thicker. All right? Which means it has a thicker, what they call the tunica media. There's three tunics that make up the vessel: the tunica intima, the media, and the adventitia. All right? The tunica intima is endothelial tissue and forms a nice barrier between the blood that the vessel carries and the rest of the body. The tunica media is smooth muscle tissue, which is designed to control blood pressure and also to engage in something called vasomotion, where we can move blood to organs and organ systems that need it most desperately when we change our activity level from, say, resting to exerting, right? And the tunica adventitia is connective tissue that latches the blood vessel to the surrounding tissue so it doesn't twist, buckle, or kink. Okay, and veins and arteries have all three of those layers. Capillaries just have an endothelial layer, a basal lamina, and then they are in contact with the tissue on the outside. All right, you can see an artery here. See how thick the smooth muscle layer is. This darker pink is the endothelial tissue of the intima. And then out here, you've got the adventitia, all the way here on the outside. In case you can't see me drawing, let's use the highlighter. And I'll use color. Let's do, I don't know, green. And I want the laser pointer. Laser color green. There we go. And laser pointer. There we go. And so that's the adventitia, right? That's the tunica media, okay? And that is the intima, okay? And the vein has the same layers, right? The intima there is just that little rim of sort of purple nucleated endothelial cells. There is a media. It's not very much smooth muscle, but you can see some of it, right? And then you've got adventitia on the outside. Now, notice that the dimensions of the vein are different. The lumens are much bigger and the walls are much thinner, and that represents two things. Number one, the blood in veins is under much lower pressure, so it doesn't exert as much force against the walls of the vessel. And in addition, it has a large lumen because it serves as a blood reservoir, right? At any moment in time, there's going to be more blood in the venous return to the heart than there's going to be in the arterial system, okay? Generally, arteries and veins travel together, right? And usually you'll also find a nerve and a lymphatic with them, and that's to maintain the structural integrity of the body, because if everything went an independent route, what would happen is that the connective tissue that holds the body together would be way weaker, right? The way I like to look at that is it's kind of like putting a new stove in your kitchen, right? A stove needs an exhaust, it needs a gas, it needs electric, right? And if you punch 3 holes in a retaining wall for each of those leads, you weaken the wall. Whereas if you do a single hole and then you put a sleeve around all those leads to the stove, then the wall is going to be way stronger, right? And so the body's designed that way too. Things tend to take common routes through the body. The lumen is the area where the blood's found in the vessel, and the layers are as we indicated, and the capillaries are just endothelia with basal lamina, and the walls are less than a micron thick, and that's so the gas exchange can happen, right? You can see here the makeup, right? In a typical artery, you've got adventitia. That's connective tissue. There's your smooth muscle, right? And there's your endothelial layer, and then you'll have in many cases an elastic lamina in between them, and that's to allow some stretchability in the artery so that you don't have such dramatic changes in pressure that you rupture the vessel, right? You don't blow it, okay? And then we eventually deliver blood to smaller and smaller arteries, and eventually we get to something called arterioles, and then they're going to empty into what are known as capillary beds. And the capillary beds are just endothelial tissue and basal lamina, tiny diameters and thin walls for gas and nutrient and waste exchange, right? And that's also why the capillaries have the greatest surface area of any of the other blood vessels in the body, right? Because they have to do a lot of exchange, and so they're very extensive, okay? As the oxygenated blood flows into the capillary bed and dumps off its oxygen and picks up its CO2 and dumps off its nutrients and picks up its waste products, it'll change color from a bright to a dark red, which looks blue through the walls of the vein. And then these capillaries will coalesce into venules, and then the venules will dump into veins. And then in the veins, again, we have the same basic layer. We don't have an elastic lamina, but we do have an unusual feature in the intima. We have valves that are made out of endothelial tissue, and they are to promote the one-way flow of blood back to the heart. All right. The reason for that is that the blood in the veins is under such low pressure that it needs help to get the blood back to the blood pump. Otherwise, we're not going to have sufficient venous return to the heart to maintain consciousness. This is also why, if you stand at attention for a long period of time, like they do in the military, you should never lock your knees, because the blood will pool in the lower extremities. You won't get enough venous return to the heart to get blood to the brain, and you'll pass out. In fact, there's some pictures you can look up on the internet of the Castle Guard over in London, you know, where they stand at attention for long periods of time, and occasionally you'll see one of them flat on his face because he was standing at attention too long and he wasn't working his leg muscles, okay? This is also why if you travel for long periods of time on a plane or a train or even in an automobile, you need to move your legs occasionally, because if the blood pools too much, the blood will clot, and then the blood clot can travel to the brain or to the heart or to the lungs, right? And you know what happens then. So the tunica intima is just endothelial layer, which is continuous with the tunica intima of the heart, okay? There's a subendothelial layer in vessels that are larger than 1 millimeter wide, and connective tissue basement membrane as well, all right, to hook up the layers to each other. The media is smooth muscle tissue that is under the control of the sympathetic division of the autonomic nervous system, which induces a feature known as vasoconstriction in the smooth muscle that supplies organ systems that aren't critical during fight or flight, such as the digestive, the urinary, and reproductive system, and promotes vasodilation in blood vessels that are critical during fight or flight, such as the musculoskeletal system, the heart, and the lungs. Okay, vasodilation just means that the blood vessel opens up, and vasoconstriction means that the blood vessel starts to crimp down, right? So when a vessel is in vasoconstriction, the pressure in the vessel goes up, and when it's in vasodilation, the pressure goes down, okay? The tunica externa is connective tissue that is going to latch the vessel in place so it doesn't twist, buckle, or kink, and that avoids the possibility of cutting off blood flow to tissues that are downstream, okay? In very, very large vessels, there are blood vessel supplies to that tunic called vasa vasorum that nourish the outer layer of the vessel, since it can't get the oxygen from the blood that it carries because that's too great of a distance. Okay, so vasa vasora are blood vessels that provide blood to other blood vessels. A little weird, but there you go. Right, vessels are going to be different in their length, their diameter, their tissue thickness, and their makeup. And you can see here how the fluid flow in the body works. Right, the heart is getting blood from the body, okay, on the right side. The heart pumps that blood to the lungs from the right side to each lung, right and left, and then the blood from the right and left lungs oxygenates and comes back to the left side of the heart into the left atrium, goes through the bicuspid valve into the left ventricle, and then the left ventricle, the most powerful chamber, pumps that blood out to the body. We hit first elastic, then muscular arteries, okay, and then finally we get to arterioles, and then we're going to empty into capillary beds. All right, some fluid will be lost at the arterial end of the capillary bed, and that fluid is mostly recovered at the venial end, right? But some, about 15%, stays in the tissue, and that gets picked up by the lymphatic capillaries, where it'll be filtered in lymph nodes and cleared of pathogens and cancer cells, and eventually will return to the blood at the subclavian veins. Okay? The remaining fluid goes back into the bloodstream. The formed elements never leave, right? The red and white blood cells and the platelets stay in the vessels. And then the capillaries condense into venules, and the venules feed into veins, and the veins are going to feed eventually back up to the vena cava, and that's going to dump into the right atrium of the heart. Okay, so that's the flow. All right. The elastic arteries are thick-walled and have elastin in all three tunics. The aorta and its major branches are elastic arteries. They have large lumens, so that there's not a lot of resistance in blood flow. And the reason that a big diameter vessel provides that is because there's less drag on the walls of the vessel when you have a wide diameter, because less of the fluid is in contact with the walls. Okay, so the fluid in the middle of a big vessel moves faster than the fluid on the edges. Okay. It's inactive in vasoconstriction, so it doesn't do much vasoconstriction, vasodilation. It's a pressure reservoir because it can expand and recoil as you eject blood from the heart because the walls are stretchy, all right? And that keeps the vessel from rupturing, okay? And that smooths the pressure changes downstream. Distal to the elastic arteries are the muscular arteries, and these deliver blood to body organs. They have a thick tunica media with more smooth muscle, and these are the major players in vasoconstriction and vasomotion. Right? These are the guys that determine your overall blood pressure to a large degree, and also determine where most of your blood gets routed, depending on whether you're in a rest and digest state or a fight or flight state. Okay, and that's all as a result of signals that come from the sympathetic division of the autonomic nervous system. Arterioles are the smallest arteries, and they feed into capillary beds and control flow into those beds by vasodilation and vasoconstriction. And most of that control is due to the chemical state of the tissue. All right, we're going to find out that in tissues that are low oxygen, high ADP, high CO2, and low pH, that they promote the opening up of those capillary beds under those chemical conditions, and that promotes perfusion. And that brings more blood into those tissues and washes away or takes away the waste products. Whereas in tissues that are well perfused, you're going to see high oxygen, high ATP, low CO2, and higher pH. And those tissues are going to be less well perfused. And what happens from moment to moment is that we switch off which capillary beds are opened up and which are closed as another way of moderating your blood pressure. At any moment in time, about 3/4 of your capillary beds are shut and 1/4 of them are open, and we switch off which are open and shut from moment to moment, right? If we were to open all your capillary beds at once and then flood them, your blood pressure would drop so low that you would pass out. You might even die, and that's a condition known as shock, right? So the capillaries are the thinnest of the blood vessels. They have the narrowest diameters. They don't really have a tunica media and not really a tunica adventitia. They just have an intima and some basal lamina. There are parasites that stabilize the walls and can control their permeability in many cases, but the diameter only allows single columns of red blood cells to pass through at a time, and they do so very slowly. And the reason for that is to give time for the gas and nutrient and waste exchange to happen. Okay, so blood flow in the capillaries is slow, but the pressures are also very low. Okay, so low and slow. Okay, in all tissues except for cartilage, epithelia, and the cornea and lens of the eye, you'll find a capillary supply. All right, now you might wonder, okay, well then, how does cartilage, epithelia, cornea, and lens get its oxygen? Because it's still living tissue. And the answer is the big D, diffusion. Okay, cartilage, for instance, is found primarily in places like the ends of the bones, the larynx, the trachea, the bronchi, and the epiglottis. Okay, and in some cases, it's surrounded by something called periosteum, which had blood vessels in it, which let oxygen diffuse into the cartilage matrix and from there to the chondrocytes, all right? But in some cases, it's bathed in synovial fluid, like the cartilage in the joints. And in that case, the oxygen diffuses from the blood vessels around the joint into the synovial fluid, and then from the synovial fluid into the cartilage. And that's what keeps it alive. And so this is one of the reasons why a moderate amount of movement is critical to keeping your cartilage healthy, because if you become relatively inactive, the oxygen supply and the nutrient supply in joint cartilage becomes poor enough that the cartilage can thin prematurely. All right. The epithelia, as you know from AP1, gets its oxygen from diffusion from the dermal layer. Okay, so the dermal layer underneath the epithelial layer has blood vessels in it. The oxygen diffuses into the epithelial tissue and keeps it alive. Remember that epithelial tissue is avascular. Okay, the cornea is a special case because the cornea doesn't really have connective tissue under it. Instead, it is a layer of epithelia, stratified epithelia, that gets its oxygen from diffusion from the air. Okay, so the oxygen for the cornea actually comes from the air in front of your eye, as opposed to a blood supply. And that's one of the reasons why if you leave your contact lenses in too long and they get protein deposits on them, you can create a wound in your cornea, an ulcer, and that is a very, very painful condition. Okay, and the lens of the eye, it's the same deal. It is diffusion. Okay, and again, through the humors and into the lens, right? The aqueous humor can allow diffusion of oxygen all the way to the lens. Okay, so again, the main function of the capillaries is to provide extensive surface area for exchange, all right, for all the things that blood carries. There are different types of capillaries: continuous, fenestrated, and sinusoidal. The most permeable are the sinusoidal, and the least permeable are the continuous, but they're all pretty permeable. Okay, continuous capillaries, where do you find them? Skin and muscle. Tight junctions connect the endothelial tissue, and there are clefts that allow some fluid movement and small solutes to go from the capillaries into the surrounding tissue. Okay, the capillaries in the brain are unique because they're associated with glial cells that cause these endothelial clefts to close up, and it's only the astrocytes that basically do the grocery shopping for the nerve cell by removing from the capillary nutrients and dropping off into the capillary waste products that can't diffuse through the walls of the vessel. Okay, the oxygen and the CO2 can still diffuse, and they do. Okay, but remember that the blood-brain barrier exists as a way of protecting the brain and the rest of the central nervous system from pathogens that are in the blood that, if they got in there, would cause a lot of trouble, because the immune function in the central nervous system is limited to the action of only the microglia, right? And they are basically, they don't have enough function to fight off a massive infection. And in fact, when you do break the blood-brain barrier, one of the things that happens is that there is invasion of immune cells, and when they get in there, they damage nerve tissue because they've never seen it before. And that's how you produce scars that don't allow nerve cells to knit back together to a large extent. Okay? So you can see a continuous capillary here. The paracytes, the endothelial cells, the basal lamina, and you can see the narrow diameter, right? One blood cell, one red blood cell width across approximately. So these guys, again, go through it like stacks of coins. And remember, since red blood cells don't have nuclei, they get a lot of their damage going through the capillaries, and that's why they only last 120 days in circulation, because they don't have much repair capability. Okay? The fenestrated capillaries have endothelial cells with pores in them and are more permeable than the continuous, and they're important for filtrate formation and absorption. So where do we find them? Small intestine, endocrine organs, and kidneys. And that's what they look like. And you can see little openings here. Okay? So you let more stuff through, right? Sinusoidal are the most permeable, right? They have large intercellular clefts and big lumens, and the flow through them is very slow, okay? Big molecules and even blood cells can pass between the blood and the surrounding tissue. We find them in the liver, the red bone marrow, the spleen, and the adrenal medulla, okay? And there are macrophages in the lining that wipe out pathogens before they get a chance to get into the tissues themselves. And that's what they look like, okay? So, you know, pretty large openings compared to the other capillaries, okay? The most permeable. Now, capillary bed is fed by a terminal arteriole that leaks into a meta-arteriole that leaks into a thoroughfare channel, which is connected to other capillaries, all right? And when we want to perfuse the capillary bed, the sphincters that guard between the thoroughfare channel and the rest of the capillary bed close, and the blood goes right through the thoroughfare channel into the venous return. And when we and that's when we want to bypass the capillary bed. When we want to perfuse the capillary bed, we open up all of those sphincters, and the blood floods into the bed, and you have exchange of oxygen and nutrients, right? The thoroughfare channel is going to drain into the postcapillary venule, and that's going to drain ultimately into a vein, right? Now, occasionally you'll have shunts, right? The meta-arteriole is a thoroughfare channel that is going to connect a terminal arteriole and venule, right? But there are other examples of shunts. There are shunts between veins that provide collateral drainage, okay? Because you have to drain from both superficial and deep parts of the body, so they have connectors running between parallel veins to go back to the heart. And then occasionally you're going to have connections between parallel arteries, all right, in special situations where a vital organ needs a blood supply, such as in the brain or in the heart. in every location with the exception of where the coronary arteries come off, right? And the reason, again, for those connections between parallel vessels is to allow blood to flow around a potential block and to keep the vital organ working. And then occasionally you're going to see an arteriovenous shunt. Okay, those are rarer, all right? But most of the time when blood goes from an arterial to a venual, it's through a thoroughfare channel in the body. Okay, true capillaries have 10 to 100 exchange vessels per bed and branch off the meta-arteriole or terminal arteriole. True capillaries branch from the meta-arteriole and return to the thoroughfare channel. And it's the precapillary sphincters that regulate the blood flow into them, right? So these are little rings of smooth muscles that respond to those chemical signals I was talking about, right? So when the ADP is high, the pH is low, the CO2 is high, and the oxygen is low, what do? The precapillary sphincters open up, and the blood flows into the bed and perfuses it like that, right? And then when those conditions are reversed, we close those precapillary sphincters, and the blood goes straight through, right? And as we talked about earlier, at any moment in time, 3/4 of your capillary beds are shut. As a result, we maintain your blood pressure, right? Venules are formed when the capillary beds unite, okay? They're small post-capillary venules that are very porous, and this allows fluid and white blood cells to go into tissues, and they have an endothelial layer and some parasites. Larger venules have a couple of layers of smooth muscle as well, okay? Veins are formed when the venules converge. They have thinner walls and bigger lumens. The blood pressure is lower than in the arteries. It has a thin tunica media, a thick tunica externa, and it has collagen and elastin networks in the connective tissue layers. And they're called capacitance vessels because at any moment in time, they carry most of your blood in your body, okay? About 65%. So you can see here at rest, right? Most of it's going to be where? In the systemic veins and the venules. The systemic arteries and arterioles, about 15%. The pulmonary circulation, 12. The heart itself, 8. And the capillaries, about 5, right? So the bulk of your blood over here in the veins, right? The other 40% over here. There are adaptations that help to make sure that we can return the blood back to the heart even though it's under low pressure. And we talked about the valves earlier and the fact that they only open towards the heart ensures their flow towards the heart. We also have some smooth muscle in the media of the veins, and that helps to squeeze the blood a little bit. But it's primarily the skeletal muscle and the respiratory pump that help to get the blood back to the heart. The skeletal muscle pump is the fact that you move, run, jump, and sit. And when that happens, muscles contract. And when the muscles contract, that squeezes blood through those valves and helps get it back to the heart. And the respiratory pump is just the pressure changes that take place in your thoracic cavity when you breathe in and out. And that allows the vena cava to change its pressure, and that can draw blood up towards the heart as well. Okay. They also have large diameter lumens, so the resistance is very low. Okay. And you can see here again how the walls of the venules, the veins, and the capillaries differ, right, in terms of percentage of wall dedicated to a particular tissue type. But keep in mind also that in every case, this represents an organ, right? Because these are different tissues cooperating to do something that one tissue can't. You've got smooth muscle, you've got endothelial tissue, and you've got connective tissue, and in many cases, nerve supply, and the blood they carry is connective tissue, and so that's an organ. You can see here again back to our picture from earlier, okay? And we talked about anastomoses, right? Interconnections of blood vessels that have different purposes, right? The interconnections between veins are designed, again, to promote drainage from both deep and superficial tissues back to the heart. The interconnections between arteries are designed to provide collateral circulation so that we don't end up um, cutting off blood flow to things like the brain and the heart, right? And then we have um, connections occasionally between arteries and veins, right? But most of those are thoroughfare channels, right? Now, we want to talk a little bit about blood flow, okay? And blood flow is essentially the physics that govern the speed with which the blood moves through the vessel, all right? We measure it in milliliters per minute, and it's equivalent to something called cardiac output for the entire vascular system. Now, this is an important point that I didn't get a chance to hit in lab straight up because we ran a little short on time, but again, it's in the podcast. But your stroke volume times your heart rate, okay, is equal to your cardiac output. All right, your stroke volume is the difference between the end diastolic volume of the ventricles and the end systolic volume, right? And your heart rate is just number of beats per minute. All right, so you take milliliters per beat, all right, which is your stroke volume, times beats per minute, okay, and you have milliliters per minute, and that's your cardiac output, all right. It's pretty constant when it's at rest. It varies in individual organs based on what the organ needs, all right. Now, your blood pressure is just the force per unit volume that the blood exerts on the wall of the vessel that it carries. It's expressed often in millimeters of mercury, just like barometric pressure is. And again, the logic behind that is that when you use a barometer, okay, that's a glass hollow tube that's closed at one end and open at the other, and it has liquid mercury in it. And basically, the mercury gets pushed down on by the weight of the air above it in the atmosphere, and in the other end of the tube, the mercury will rise, and you can measure the number of millimeters of mercury, and that'll be proportional to the atmospheric pressure, right? That's where that measurement comes from. So 760 millimeters of mercury is equal to what we call 1 atmosphere, right? In the body, we can measure it as pressures, right? We measure systemic arterial blood pressure in large arteries that are near the heart, and the pressure gradient is what drives the movement of the blood through the system, okay? So what fights that movement, all right, is resistance, right? Flow is proportional to the difference in pressure between 2 points in the system divided by the resistance. So the greater the resistance, the less the flow, and the greater the difference in pressure, the greater the flow, all right? So if you've got a high resistance vessel, very little flow is going to happen, all right? If you have almost a zero pressure difference between 2 points in a system, a fluid system or a gas system, then the net movement is going to be zero, all right? So the flow will be virtually non-existent, okay? So what provides resistance? The viscosity of the blood, which is the thickness of the blood, the length of the blood vessel, and the diameter of the blood vessel. Now, how do these affect it? The higher the viscosity, the greater the resistance. The longer the blood vessel length, the higher the resistance. And the smaller the diameter, the higher the resistance. The greater the resistance, the less the flow. The less the flow, the greater opportunity there is for the blood to clot. All right, so just keep that in mind. The factors that remain pretty constant in our body are our blood vessel length and our blood's viscosity. Okay, the body has systems that are designed to make sure that those stay within limits compatible with life. All right. So what changes from moment to moment in order to adjust our blood pressure as environmental conditions change is the diameter of the blood vessel, because that has the greatest influence on the force against the wall of the vessel, because it varies with the fourth power of the vessel radius. Meaning what? All right, if we take blood vessel X, all right, and we reduce the radius by half, the pressure is going to go up by 2 to the fourth power. So 2 times 2 is 4, times 2 is 8, times 2 is 16. So the pressure will be 16 times greater, okay? So keep that in mind, right? If you change the diameter of the vessel, you have a huge influence over the flow and over the pressure, all right? And that's part of what vasomotion is about. By increasing resistance to less necessary systems and decreasing resistance to more necessary systems, we can route blood, for instance, away from the urinary, digestive, and reproductive system and to our heart, lungs, and skeletal muscle when we go into things like fight or flight. Okay? All right. Small diameter arterioles are the major players in peripheral resistance. Big changes in the diameter of fatty plaque from atherosclerosis can also increase the resistance by causing the flow to encounter friction, turbulence, right? And that irregular movement increases the resistance to the point where flow through that region slows and you can generate now blood clots, all right? So this is one of the reasons you want to keep the amount of saturated fat and cholesterol in your diet minimized, because that contributes to the formation of these plaques that collect on the walls of the arteries, and that slows the blood flow down, and that makes it more likely you're going to generate a traveling clot, which is called an embolus, which could jam in a blood vessel called a thrombus, and that'll cut off blood flow to the tissues, and they'll die. Now, frequently, when you have conditions like atherosclerosis, you also have what's called a comorbidity known as obesity, meaning you're heavy, all right? And when you're heavy, your blood vessels also change in another way. They get longer. And when the blood vessels get longer, the resistance also goes up. So when you combine atherosclerotic plaque with a heavy body, then you're really putting a lot of pressure on the heart, and it can get to the point where the heart is trying to pump against so much resistance that it can't open the semilunar valves anymore. And that's a condition known as congestive heart failure, and that's lethal in about 6 or 7 minutes, okay? Now, when you put on good weight, muscle weight, something different happens. You actually produce tissue that causes more blood vessels to grow. And so you reduce the resistance on the heart, because now you have more places for the blood to flow with each beat. And what that means then is that the heart can pump low and slow and still maintain perfusion without having to work so hard. And that's why in well-trained athletes, regardless of whether they are exercising or whether they're at rest, their pulse is going to be low and slow. Their heart rate is going to be low and slow, and their pressures are going to be low, okay? And that's one of the major payoffs of exercise. So we talked about flow and resistance, right? That's the equation. Delta just means the difference between, right? So you got two locations, location 1 and location 2. The difference between pressure at location 1 and pressure at location 2 would be that quantity, okay? So the higher the difference in the pressure and the lower the resistance, the greater the flow, okay? So resistance is more important in influencing local blood flow because we can change it by doing vasodilation and vasoconstriction. Now, the pumping action of the heart is what provides the blood flow, right? The pressure results when the flow is opposed by resistance. Systemic pressure is going to be highest in the aorta and then drop as we move through the pathway, reaching about 0 millimeters of mercury in the right atrium, which is where the blood gets dumped back into the right upper chamber of the heart. Okay. The steepest drop is going to occur in the arterioles, and you can see that here. And you can also see that the greatest pressure differences are going to be near things like the aorta, right? And the smallest pressure differences are going to be in the venous return. And this is why when you take a pulse, you have to do it in an artery, and also why you have to take a blood pressure in an artery, right? Because the pressure changes in the veins and the venules is too small to detect or to palpate, right? And that also brings up an interesting point, which is that the areas on your body which are pulse points, such as your radial pulse point on the right side of either wrist by the thumb, right? Right side of the right wrist and the left side of the left wrist. Um, the pedal pressure in the vessels that run across the dorsum of the foot, the top of the foot, the pressure in the brachial artery, right? Those are all places where you can take a pulse. You can also do it in your carotid artery, which supplies blood to your brain, and it runs through your neck. And those are all places where you can feel the pressure differences fairly easily, okay? They also happen to be places not only where you can take a pulse, but in places other than your neck, you can take a blood pressure, okay? And that allows you to determine the state of the system, okay? If you have a blood pressure that's consistently high, that's a condition known as hypertension, and that often is related to things like obesity, all right, and age. And the danger, of course, with hypertension is that the pressures can get so high in the whole system that you can blow out capillary beds and produce a stroke or a heart attack, okay? If the pressures are too low, then we have inadequate perfusion, and that can result in tissue death, right? And that's less common, but it does happen, okay? The arterial blood pressure is going to reflect the elasticity and the volume of the blood that enters those great vessels, okay? Blood pressure in the heart is pulsatile, which means it has both a systolic and the diastolic component. The systolic pressure is the pressure when the ventricles are squeezing, and the diastolic pressure is the pressure when the ventricles are relaxed, okay? So when you take a blood pressure, it's always systolic over diastolic, okay? The pulse pressure is the difference between the systolic and the diastolic. So you subtract the diastolic from the systolic, and you get the pulse pressure. Now, why do we care? We care because these are all indications of things like hypertension or hypotension, okay? If you have a blood pressure 120 over 80 or higher, you have hypertension, right? And that means you need to take steps dietary and exercise activity-wise to try and reduce those pressures, okay? Otherwise, you're going to put yourself at risk for a heart attack or a stroke, okay? The mean arterial pressure is what pushes the blood to the tissues, and it's 1/3 the pulse pressure plus the diastolic pressure. So in a typical, let's say 120 over 80 was somebody's reading, right? Then the diastolic pressure is 80, right? 1/3 the pulse is going to be what? Well, what's 1/3 of 80? Well, 80 divided by 3, all right, is going to give you around 13, all right? So the mean arterial pressure is going to be 93. And again, it's something that can indicate things like hypertension. Capillary blood pressure is typically low, ranges from 17 to 35 millimeters of mercury, and that low pressure is important so that you don't blow the capillary beds, and you give time with that low pressure and slow flow for exchange to take place, and that's what the capillary surface area is for. Venous blood pressure doesn't change much during the cardiac cycle. There's a very tiny pressure gradient, about 15 millimeters of mercury, and low pressures because of the cumulative effects of peripheral resistance. So the energy of the blood pressure, most of it is lost as heat and work as the blood passes through our circulatory system, right? So under these low pressures, we need help to get the blood back to the heart, and that's where the muscular pump and the respiratory pump and venoconstriction come in, right? The muscular pump is just the fact that you move, and as you move, that squeezes blood through valves and back to the heart. The respiratory pump is the fact that you breathe, and those pressure changes in the chest cavity milk the vena cava and pull blood back up towards the heart, and the venoconstriction is just the smooth muscle action, right? And so there's the muscular pump, right? You push on the sides of the vessel, you force the blood through the valves, and it slowly gets back to the blood pump, okay? So the more active you are, the more venous return you have. The more venous return you have, the more blood the heart will pump out, and that's going to be better for you for a lot of reasons, okay? That's going to help with filtering pathogens out of the fluid that's in the blood because it'll deliver more fluid to the lymphatic system. It's going to be good because you'll supply more oxygen and nutrients to tissues that need it, okay? And it's going to be good because you're going to build lean muscle mass and burn fat. And that's going to take the resistance off of the heart and allow it to beat more effectively, okay? To maintain blood pressure, we need the cooperation of the heart, the blood vessels, and the kidneys under control of the nervous system. And there's also some hormonal control as well. The main factors are the cardiac output, the peripheral resistance, and the blood volume, okay? How so? Higher the cardiac output, the higher the pressure. The higher the peripheral resistance, the higher the pressure. The higher the blood volume, the higher the pressure, okay? So if you can look at these equations up top, right, we know that the flow is equal to the difference in pressure over the resistance, right? The cardiac output is equal to the difference in pressure over the resistance. And so the difference in pressure is equal to the cardiac output times the resistance, right? We just rearrange this equation here, okay? The blood pressure is equal to the cardiac output times the peripheral resistance, and the cardiac output also depends on the blood volume. The blood pressure varies directly with the cardiac output, peripheral resistance, and the blood volume, so changes in one is usually compensated for by changes in the other, okay? Cardiac output, remember, is equal to the stroke volume times the heart rate, with a normal cardiac output at rest being between 5 and 5.5 liters a minute. It's determined by the venous return and also the endocrine and the nervous system, specifically the autonomic nervous system. Resting heart rate is maintained by the vagal inhibition of the intrinsic heart rate from the vagus nerve. So what the vagus nerve does is it serves sort of as a braking mechanism for the intrinsic heart rate, which is about 100 beats per minute, okay? And that's the intrinsic rate of the SA node firing. Our normal rate is about 75, okay? So if you were to cut the vagus nerve to the heart, what would happen is the heart would speed up quite a bit, okay? The stroke volume is controlled by the venous return, right? The more blood that goes into the heart, the more blood comes out of the heart. Now, that might seem obvious, right? Because if that weren't the case, if you were throwing more blood into the heart than the blood would pump out of the heart, what would happen to the heart? It would explode, okay? But there's a reason that the more blood you throw into the heart, the more it can squeeze out. You'll see that in part B of the heart chapter in the podcast, but I'll tell you now that basically when you stretch the ventricle walls out enough, you get more power from the myocardium. The gripping and the squeezing is better, and so more blood can shoot out into the pulmonary trunk and into the aorta. So more out, more in, more out. That's called the Frank-Starling law of the heart, okay? During stress, the cardioacceleratory center is going to increase your heart rate and stroke volume through sympathetic stimulation. Remember that the autonomic nervous system is a two-neuron pathway between the central nervous system and the target. There's a pre and a post-ganglionic neuron, all right? And in the sympathetic division of the autonomic nervous system, the preganglionic neuron hits something called the sympathetic chain ganglia, and then a postganglionic neuron carries the information on to the target, right? So it's the postganglionic neuron that's going to talk to the heart, and the neurotransmitter it uses is something called epinephrine, all right? And so what happens when epinephrine hits the targets on the heart, like the SA and the AV node, is that the end systolic volume goes down and the mean arterial pressure goes up. So it's another way of saying that the heart squeezes harder, okay? In addition, the venous return to the heart goes up, right? So more blood in, more blood out, right? And the heart rate goes up as well, okay? So let's see how exercise does its magic, right? Exercise, right? Your skeletal muscle starts to do its work. You start to breathe harder. Sympathetic venoconstriction kicks in because of the anticipation of the exercise. That's coming from your cerebral cortex's influence on the hypothalamus and the brainstem, which are the master control centers of the autonomic nervous system. Okay, that's going to result in an increased venous return and an increased end diastolic volume. More in, more out, right? Because the stroke volume is going to get bigger, because the end systolic volume is going to drop, because it's going to squeeze more blood out of the heart with each stroke. Why is that happening? Blood pressure drops, activate cardiac centers in the medulla. Sympathetic activity squirts epinephrine into the blood from the adrenal medulla and also from the postganglionic sympathetic nerve endings. The result is the heart rate goes up and the contractility goes up, so the heart beats quicker and beats stronger, right? And the result is that cardiac output jumps quite a bit, actually, okay? Short-term neural and hormonal controls are going to counteract changes in blood pressure by changing peripheral resistance and cardiac output. Long-term renal regulation is going to deal with changes in fluid volume, okay? So the neural controls maintain MAP by changing blood vessel diameter, vasoconstriction, vasodilation, vasomotion, okay? If there's a low blood volume, all the vessels will constrict, except those that go to the heart and the brain, because those are your vital organs. And this is going to change the distribution to the organ systems in fight or flight, okay? So what are the controls that operate here? Well, they're called reflex arcs, and they work through baroreceptors that can detect things like atrial stretch, okay, and aortic stretch. There's aortic and carotid bodies that are able to pick up stretch, okay, in the great vessels and also in the atria, okay. The cardiovascular center of the medulla oblongata responds to the input from the vasoreceptors and the vasomotor fibers to the heart, and vascular smooth muscle gets stimulated, and there's sometimes input from chemoreceptors and other brain centers, like when you panic, for instance. Sympathetic neurons in the medulla are going to look at changes in cardiac output and blood vessel diameter and adjust them. They consist of cardiac centers and the vasomotor center, which sends impulses through sympathetic postganglionics to blood vessels and moderates constriction, and that's what we call vasomotor tone. And it receives input from baroreceptors, chemoreceptors, and higher brain centers, okay? And when we come back next week, the maintenance of blood pressure and blood flow is critical for proper functioning of the circulatory system. And we talked about some of the factors that regulate blood pressure. One of them is the length of the blood vessels. One of them is the diameter of the blood vessels. And then there is the cardiac output, all right? And then finally, there's the volume of blood in the system, okay? Now, if we look at baroreceptor mechanisms. Baro means pressure, right? And there are pressure receptors that are in the carotid arteries, which bring blood up to the brain, in the aortic arch, and in the walls of the large arteries of the neck and the thorax. And what they do is they detect changes in pressure in those vessels and send commands in order to keep the blood pressure within a window compatible with normal function, right? If the blood pressure gets too high, you run the risk of blowing capillary beds and cutting off blood to everything downstream. And if the pressure gets too low, then you don't have enough push behind the fluid in order to perfuse the capillary beds in order to keep everything functioning normally. Okay, so we have to keep our blood pressure within a window. Remember that blood pressure is typically measured by a sphygmomanometer. A normal blood pressure on average is about 120 over 80, where 120 would be the systolic pressure when the ventricles are squeezing, and 80 would be the diastolic when the ventricles are relaxing. Okay. On these baroreceptors, when the blood pressure goes up, all right, that causes the baroreceptors to fire, and that sends input back to the cardioacceleratory centers in the brainstem. And what that'll result in is nerve output that promotes dilation of arterioles and dilation of venules and veins as well. Okay. And what that does is that causes the container in which the blood sits to get larger, and that causes the pressure in the circulatory system to go down. Right. It also stimulates the cardioinhibitory center, and that causes the rate and the force of the contractions of the heart to be reduced. And all of that is going to reduce our blood pressure. So the decrease in blood pressure, again, venodilation, arteriolar vasodilation, drop in cardiac output. Okay, and remember what cardiac output is the heart rate times the stroke volume, right? The heart rate is beats per minute. The stroke volume is milliliters per beat. If the mean arterial pressure is low, we have reflex vasoconstriction, an increase in cardiac output, and an increase in blood pressure. An example, when you stand up, baroreceptors in the carotid arteries protect blood flow to the brain. In the systemic circuit as a whole, the aortic reflex maintains your blood pressure. All right. In fact, sometimes if you stand up too quickly, and especially if you're older, you can have syncope, which is fainting. Right, and that's where this carotid sinus reflex isn't working as effectively as it otherwise would. Right, because we need to keep blood flow to the brain constant in order to remain conscious. Okay. These become ineffective if an altered blood pressure is sustained. So if you're always hypertensive or always hypotensive, then the baroreceptors are going to be outside their area of effectiveness, will be beyond their window to control blood pressure. So you can see an example of how the flow works here, right? So if we turn on the laser pointer, okay. Let's take this. Here we have homeostasis, right? Everything's normal. Then we have blood pressure rising, baroreceptors firing, brainstem picks that up, cardioacceleratory center is inhibited, cardioinhibitory center is stimulated, the rate and force of contraction of the heart go down, so the cardiac output drops, and we see vasodilation, and that drops the blood pressure down to where it should be. Once we're back in homeostasis, if our blood pressure, say, were to continue to fall, then baroreceptors would pick that up. They would signal the cardioacceleratory center in the brainstem. That would increase the cardiac output and cause vasoconstriction. Okay. There's also chemoreceptors in the aortic arch and in the carotid arteries that pick up changes in CO2 concentration or a drop in pH, which are really two ends of the same thing. Remember that when CO2 dissolves in water, it forms carbonic acid, and that causes the pH to drop. Okay, so you're really talking about the same phenomenon here. The oxygen change it's important, but it's not nearly as critical in determining the rate and force of contraction and the maintenance of blood pressure as CO2 is. Okay, and that's because CO2 dissolves much more easily in water than oxygen does. Okay, the cause of increased blood pressure by signaling the cardioacceleratory center makes the cardiac output go up. So the rate and force of contraction of the heart go up, cardiac output goes up, so the motor is running hotter, the pressure goes up. The vasomotor center is going to promote vasoconstriction, and that's just like shrinking the container in which the blood is located, and that's going to cause an increase in pressure as well. In the medulla, the hypothalamus and the cortex can modify blood pressure through relays to the medulla. So, for instance, when you get anxious, right, you perceive that in your conscious mind, and that can act through the brainstem to cause your blood pressure to go up. And I'm sure you've seen people who are anxious or angry have the blood vessels in their head pop out, right? And that indicates a physiological change to deal with stress, right? Hypothalamus is going to increase blood pressure during stress and is going to control the redistribution of blood flow when you're in a fight or flight situation. And this is primarily the result of the neurotransmitter slash hormone epinephrine, also called adrenaline, right? So what are the effects of fight or flight? Well, if you remember back to ANP1 and the sympathetic division of the autonomic nervous system, you'll recall that in a fight or flight situation, release of adrenaline slash epinephrine, okay, and its cousin noradrenaline and norepinephrine is going to promote release of additional epinephrine from the adrenal medulla, promotion of vasodilation to the heart, lungs, and skeletal muscle, vasoconstriction to the digestive, urinary, and reproductive systems. It's going to promote an increase in the rate and force of contraction of the heart, okay? And it's going to promote vasodilation of vessels near the surface of the skin and an increase in sweat production in order to control the extra heat generated by the body. It also is going to promote vasodilation in the lungs and bronchodilation in the lungs in order to promote more loading of oxygen into the blood as well. And what this does is it prepares you for battle, all right? It gets more oxygenated blood to the heart, lung, and skeletal muscle more quickly, and the result is you respond more effectively to your situation, okay? And so that's the job here of the sympathetic division of the autonomic nervous system, which is run by the brainstem and the hypothalamus, right? Remember that those are sort of like your second brain. They are the control center for all the stuff that goes on in your body that you don't consciously operate. So without your hypothalamus and brainstem, you'd be dead, okay? Okay, short-term regulation is the result of changes in peripheral resistance in the system, which means a change in the force against which the heart has to work. Long-term regulation is through changes in blood volume, all right? So how much fluid is in the system. So epinephrine and norepinephrine from the adrenal gland and from sympathetic stimulation cause an increase in cardiac output and vasoconstriction. In addition, we also set in motion something called the renin-angiotensin aldosterone mechanism. And the way this works is that when the kidneys sense a drop in oxygen levels or in blood pressure, they release a hormone called renin, okay? Renin converts angiotensinogen into angiotensin 1, and in the lungs, angiotensin 1 is converted by ACE, which is angiotensin converting enzyme, to angiotensin 2. All right. The result is that we end up producing more aldosterone and more antidiuretic hormone, and that causes us to retain more fluid, and that makes the blood volume go up. It also promotes thirst. It also um, is going to promote vasoconstriction. All right, and so all of that is going to raise your blood pressure up. On the flip side, atrial natriuretic peptide is stimulated by atrial stretch, which means that the atria are over-distended, and if they're over-distended, the way that they'll respond is by kicking out this hormone that results in increased urine production and more fluid loss, and the result of that is for the blood volume to go down, okay? So you can see the renin-angiotensin-aldosterone system here, right? Remember that, well, we haven't really talked about antidiuretic hormone yet, but antidiuretic hormone is targeting the kidney tubules and causing the kidney to retain more fluid, okay? It's an antidiuretic, meaning it makes you less likely to pee. Aldosterone does this by causing increased retention of sodium by the kidneys, and the water follows the sodium passively back into the blood, and the blood volume goes up and the blood pressure goes up, okay? And then you increase water intake by increasing thirst, and then you also turn around and do some vasoconstriction, and the result is an increase in the blood pressure, right? And then a drop in arterial pressure, decreased filtration, decreased urine formation, increased blood volume, increased mean arterial pressure, right? So we make less pee, right, through the direct renal mechanism. At the same time, the renin-angiotensin-aldosterone system is working. Baroreceptors are going to adapt quickly to chronic high or low blood pressure, so they're ineffective, but long-term mechanisms are going to work through the kidneys, which have a arterial blood pressure regulation through a direct renal mechanism and an indirect mechanism. That's the renin-angiotensin-aldosterone system we were talking about earlier, right? The direct renal mechanism is just a matter of pressure, right? The less pressure in the glomerulus, the lower the GFR, the lower the amount of urine you make, okay? GFR is the glomerular filtration rate. The glomeruli are where the blood is filtered and the fluid is collected, which will eventually become urine, right? And so with the direct renal mechanism, more pressure on that filter pushes more fluid into the kidney tubules, and that's going to result in more fluid in your pee, right? And that's going to bring your blood volume and your blood pressure down. And on the flip side, if your blood pressure drops, you want to hold on to more water, so we'll slow down or we'll reduce the pressure in the glomerulus, we'll retain more fluid, and your blood volume and blood pressure will go up, okay? So that's the direct renal mechanism, right? Alters the volume independently of hormones as a result of changing the amount of urine you make. The indirect mechanism we talked about, right, which is the renin-angiotensin-aldosterone system, right? Again, to review, a drop in blood pressure, kick out of renin, angiotensinogen becomes angiotensin 1 because of the action of renin. Angiotensin 1 in the lungs through ACE becomes angiotensin 2, and angiotensin 2 is the major player here. He causes kick out of ADH, kick out of aldosterone, vasoconstriction, thirst, and all that jacks your blood pressure up. And that's one of the reasons why in people who are hypertensive, we'll frequently give them a class of drugs that are called ACE inhibitors. Okay, and what ACE inhibitors do is they cut down on the production of angiotensin 2, which cuts down on the mechanisms that elevate blood pressure, and it causes the blood pressure to drop. Okay? All right, so that's what angiotensin 2 does, right? We've already really discussed this. And you can see here the whole map again, right? The direct renal mechanism and the renin-angiotensin-aldosterone system working together to control fluid volume, right? So this is what long-term regulation looks like, not small fluctuations in blood pressure, but major ones. All right, and we need to call in all of this extra artillery in order to maintain your blood pressure in both the short and the long term, right? And so here you're just looking at factors that increase blood pressure, right? And all of these we've kind of already talked about. We could go from left to right here, right? Increased activity of the muscular and respiratory pump means more venous return, which means higher stroke volume, higher cardiac output, okay? And also because of epinephrine, the heart rate and the contractility go up, so the cardiac output rises, right? The other thing that epinephrine, of course, does is causes vasoconstriction to non-essential systems like reproductive, urinary, and digestive, and vasodilation to other systems like heart, lung, and skeletal muscle. And the result is that the pressure in the system goes up, right? Other factors, right? A decrease in the release of atrial natriuretic peptide causes more water to be retained by the kidneys, which jacks up blood volume, which jacks up venous return, which is going to jack up the stroke volume and jack up the cardiac output, right? Then we have fluid loss from hemorrhaging or sweating. Well, what's going to happen? You're losing fluid, so you got to retain as much fluid as you can. The stimulus is going to be a drop in blood volume and pressure. Baroreceptors are going to fire. We're going to see activation of vasomotor and cardioacceleratory centers in the brain. Cardioacceleratory center, what's that going to do? Jack the cardiac output. In addition, we're going to see vasoconstriction, and the result is blood volume and pressure are going to go up, okay? Primarily blood pressure here. Crisis, exercise, trauma, increase in body temperature. Stimulus, decrease in blood pH, decrease in oxygen, increase in CO2. The chemoreceptors come into play, and we kick on the vasomotor and cardioacceleratory centers. Here, an increase in vasomotor tone as a result of epinephrine or antidiuretic hormone or angiotensin II causes the diameter of the blood vessels to decrease. That increases peripheral resistance, and that increases your blood pressure. Hematocrit goes up because of dehydration. The blood viscosity goes up, the resistance goes up, the pressure goes up. Okay, this is one of the things that happens, for instance, when you donate, or not when you donate, when you get testosterone shots, is that one of the things testosterone does is elevate your hematocrit, which jacks up your blood pressure, which means occasionally you need to go donate blood so that your crit doesn't get too high, which would cause your blood pressure to get too high. And then an increase in body size, we already know, right? You get the blood vessels longer, the resistance goes up, the resistance against which the heart has to work increases, and a result is that the pressure goes up because the heart has to beat faster in order to, and with more force, in order to compensate for the back pressure, right? In order to overcome it. And that's what we call, and if that goes to its logical extreme, that's what we call congestive heart failure, where the heart has so much resistance that it's working against that it literally can't pump the blood anymore. And that's what happens a lot of times in morbidly obese people. Okay. So with that, we're done with part A of the vessels, and let's go into part B, okay, which is kind of short. So we'll start up here. Oops. Let's see. Let's save that. And we want part B. Okay. So continuing on, right? One of the things that you do typically with a patient when he comes into the office is you take his vitals, right? Blood pressure, temperature, respiratory rate, pulse, okay, are indicators in many cases of disease. So what will happen with the pulse, right, is that you're going to generate pressure changes in arteries close to the surface that you can feel with your fingers. And that's a reflection of the heart rate. Okay. The radial pulse is taken on the lateral wrist, and that's one of the routine places to do a pulse, but there are other places as well, as we'll see in a minute. And then there are pressure points that you use when you do a pulse where arteries are close to the surface and they run over bony structures, and that's why you can palpate those points. And there are the pressure points, right? Temporal artery, facial, carotid, brachial, right there at the pit of your elbow, or just above the pit of your elbow, radial on the lateral wrist, femoral in the inguinal region, popliteal back of the knee, posterior tibial on the medial ankle, and dorsalis pedis on the top of the foot. Now, not only are those places where arteries come close to the surface and you can feel the pressure changes, but there are also areas where if you get injured, you're going to lose a lot of blood really fast, okay? Because the pressure in blood in the artery is very high, and the result is that the hemorrhaging is massive. And that's why, generally speaking, okay, with the exception of the pressure points, arteries tend to run deep in the body, and veins tend to run both deep and superficial, right? Because if you cut a vein, you will lose blood, but not nearly as much. Because of the lower pressure, you have more time to set up a clotting cascade and to stop the hemorrhaging from happening, right? And that way you don't bleed to death, okay? The way that we deal with blood pressure is using something called a sphygmomanometer, all right? Basically, you have a blood pressure cuff, and then you have a dial attached to it, and the cuff can be inflated or deflated, right? So the idea with the sphygmomanometer, which is literally the dial attached to the cuff, is that you crank the pressure in the cuff up high enough to cut off blood flow in usually the brachial artery, and then you slowly release that pressure until you can hear a periodic whooshing sound, which would be the systolic pressure. You record the reading on the sphygmomanometer where that happens, and then you continue to release it until you hear no more pulsatile sound, and that's the diastolic pressure, okay? So let's take a look at how that's done. And I'll tell you that a lot of times, especially in clinical settings today, they don't use the traditional old-fashioned sphygmomanometer, right? What they do instead is they use an automated unit that records everything for you, okay? But it's still a good skill to try and master.
Before you begin, be sure to note whether the patient is sitting, standing, or laying down during the pressure reading. The standard position is with the patient sitting, with the patient's arm relaxed. However, this position is not always practical in an emergency situation. First, choose the correct sized cuff for the patient's arm. Put the cuff around the upper arm, with the cuff mark at the medial side of the biceps brachii, about an inch above the anterior elbow. Next, with your first two fingers, feel or palpate for a pulse on the radial artery. The radial pulse is found on the thumb side of the forearm at the wrist. Now, squeeze the bulb to inflate the cuff until the pulse can no longer be felt. The pressure in the cuff matches the pressure needed to close the vessel and to stop the flow of blood. Keep inflating for about 30 millimeters of mercury above the point where no pulse is felt. Place the stethoscope just under the distal part of the cuff over the brachial artery. Slowly release the pressure at about 2 millimeters per second. As the pressure in the cuff drops, blood begins to pulse through the vessel. When you hear 2 consecutive beats, record this pressure reading as the systolic pressure. Keep releasing the pressure slowly until you can no longer hear the sounds in the stethoscope. The cuff pressure reading at the time when the sound disappears from the stethoscope is called the diastolic pressure. This represents the pressure exerted on the walls of the arteries when the heart is at rest. Open the valve to deflate the cuff completely, but leave the cuff in place to save time for sequential readings. Record your information as soon as possible to avoid forgetting it. Be sure to repeat the readings over time to check for changes in blood pressure as the patient progresses. The inflated cuff compresses the tissues and vessels to stop the arterial pulse. As the air is released from the cuff, the blood vessel opens just enough to allow the high-pressure blood to pass through the vessel. As this blood passes, the partially compressed cuff creates turbulent blood flow. This turbulence can be heard by placing a stethoscope under the distal end of the cuff. At this pressure, we are reading the systolic pressure. As you slowly deflate the cuff, there is less compression of the vessels, and the blood flow becomes less turbulent, so the sound is no longer heard. The diastolic pressure represents the pressure exerted on the walls of the arteries when the heart is at rest. Remember to fully deflate the cuff when finished. Before you begin, be sure to note whether the patient is sitting.
Okay. Now, I'll tell you guys that my hearing is so poor that I can't pick up those sounds anymore. Sounds of carotid cough are below my hearing limit. So I sort of cheat the dial. And what I do is I put the cuff on the arm and pump it up and then release the pressure and watch the dial. And where the dial first starts to kick is approximately the systolic pressure. And where it stops kicking is approximately the diastolic pressure. Now, you should never do that in a clinical setting, right? You should do it by hearing when the sounds start and when they stop, or by using an automated unit, okay? So that's the systolic pressure, right? That's when the ventricles are squeezing, usually 120 millimeters of mercury, and the diastolic around 80 when the ventricles are relaxing, okay? Now, again, why do we care about blood pressure? We care about blood pressure because if it's consistently high, you run the risk of blowing capillary beds. And if it's consistently low, you run the risk of not having enough force to deliver the blood to the capillary beds. Okay? Small changes that occur during changes in posture or physical exertion or emotional upset will result in changes in your blood pressure, right? As well as longer-term changes that are a result of your age, your sex, weight, race, mood, and posture, all right? So how do these play in? Your age. The older you are, the higher your BP, okay? Your sex. Males tend to have higher blood pressure, okay? And that's because they're bigger, okay? Their cardiac output tends to be higher, and especially if they have a weight problem in either sex, when the blood vessels get longer as a result of gaining bad weight, the result is that the resistance goes up to the point where the heart has to work harder to pump, and the pressure goes up as well, all right? So that's for either sex, men or women. If you put on a lot of bad weight, your blood pressure is going to go up, right? The other thing about the difference in sex is that the testosterone increases the hematocrit, and that increases the blood pressure as well, okay? African Americans tend to have higher blood pressures, and that can be linked to things like diet, okay? Trained athletes of any race or sex tend to have low blood pressures. Why? Because they have mostly lean body mass, and their number of blood vessels is higher than in somebody who is not in good physical shape. Okay? Your mood, how does that affect your blood pressure? Well, when you're angry or panicked, your blood pressure goes up, and your posture will change your blood pressure. It'll be different when you're lying down versus when you're standing or you're sitting. Okay. Hypertension is a sustained elevated pressure of 140 or over 90 or higher. You're pre-hypertensive if your values are higher than 120 over 80, but not yet in the 140 over 90 range. Sometimes you'll have high blood pressure as a result of small changes during things like fever or physical exertion or emotional upset. That's normal, okay? But if it's always pegged within that range, 140 over 90 or higher, then you are hypertensive, okay? The reason that you see this in obese people is because the length of the blood vessels causes the resistance on the heart to go up, and the heart has to pump harder and faster to get the blood around the body, in addition to the fact that usually in obese people, you also have a condition called atherosclerosis, which causes plaque on the arteries to make the blood vessels narrower in areas, and that causes the volume of the container in which the blood sits to go down, and the pressure goes up. And a lot of times you also see in people who are obese type 2 diabetes, which means that there's more sugar in the blood than should be there, and the result is that more fluid goes into the blood, and the blood volume goes up, and the blood pressure goes up. Okay. Prolonged hypertension is a major cause of heart failure, vascular disease, renal failure, and stroke. The heart has to work harder, the myocardium gets bigger, it gets weak, it gets flabby, and it also accelerates atherosclerosis. Okay. So again, a big heart is not a good thing, right? That's called cardiac hypertrophy, and the result of that is that the heart is less effective at doing what it is supposed to do because it's overstretched, okay? And remember, in muscle tissue, that the amount of stretch before a contraction determines the overlap of the actin and the myosin and the force of the contraction. So if the heart's stretched out all the time, and it can't squeeze nearly as well, okay? Most hypertension is what we call primary hypertension, okay? And that comes from primarily lifestyle problems, right? Heredity, diet, obesity, diabetes, and stress and smoking all can contribute, right? So this is one of those things that there's both a genetic and a environmental component to. You can control this, all right, with drugs and with changes in behavior and diet, all right? Things like restricting salt, fat, cholesterol are going to help. Increase in exercise, losing weight will help. The reason, again, that exercise and losing weight will help is because you'll make more blood vessels and you'll reduce the what we call the afterload, which is the force against which the heart has to work. Okay, stopping smoking is good because that cuts down on nicotine, which promotes an increase in the heart rate. Okay, antihypertensive drugs can aid as well. Secondary hypertension usually is going to be the result of things like kidney disorders or obstructed arteries, okay, as well as things like hyperthyroidism and Cushing's. Remember that the kidneys have a big role in controlling blood pressure through two mechanisms, right? There's that direct mechanism where we make more urine when your pressure is high and less when your pressure is low. But there's also the renin, angiotensin, aldosterone system that the kidneys are responsible for. So if you have kidney disease, you have trouble controlling that, right? Okay, how about endocrine disorders? Hyperthyroidism means you make too much thyroid hormone. And the result of that is your basal metabolic rate goes up, which means your heart rate goes up, which means your cardiac output goes up, which means your blood pressure goes up, okay? Why does this happen? Well, sometimes people have cancer in the thyroid and it overproduces the thyroxine. Sometimes people get their thyroid overstimulated through autoimmune interaction, little antibodies that basically attach to the thyroid gland and latch it in the on position, such as it happens in Graves' disease. And then there's Cushing's syndrome, okay? And in Cushing's, what happens is we end up with too much sugar in the blood because we have a problem controlling cortisol. and that causes more fluid to go into the blood, and it causes the blood volume and the blood pressure to go up. Okay, so what we do here is we try and fix whatever is making that happen, and then the pressure drop will follow normally. Okay, hypotension is pressures below 90/60. It's only a problem if you don't have enough blood flow to tissues. Okay, and it can be, in its mild form, something that promotes long life and a lack of cardiovascular issues. An area where hypotension is life-threatening would be something like massive hemorrhage, right? If you're bleeding, then you're losing so much blood volume that even the corrective mechanisms can't maintain flow, and the result is that tissue begins to die, right? Orthostatic hypotension is a temporary drop in your blood pressure when you stand, okay? And that's a result of a problem with baroreceptor activity, while chronic hypotension is the result sometimes of low protein in your diet. You don't have enough protein in your diet, or you have problems with your liver, and the result of that is you don't make enough albumin, and your blood loses its volume to the surrounding tissues, and they swell up, okay? It's also a problem with Addison's disease or hypothyroid. And then acute hypotension, as we discussed, is like circulatory shock, where you're bleeding, okay? Tissue perfusion is important because it controls the amount of oxygen and nutrients that you get to the tissues to keep them alive, as well as the rate at which you remove the waste products, right? You have to do both, right? If you don't get the oxygen and the nutrients in, the tissues can't function and they can't build new stuff. And if you don't remove the waste products, you create toxic conditions that kill them, okay? So what the blood flow is doing is bringing in the goodies and taking out the trash for all the tissues in the body. So this is critical that we control the amount of blood that goes to different tissues based on their need, all right? Gas exchange in the lungs is important. The more perfused the lungs are, the better the gas exchange is. Perfusion of the capillaries in the digestive system is critical for the absorption of nutrients. And obviously the perfusion of the kidneys is what allows us to clean the blood, okay? You can see here the difference in where the blood goes depending on where we're exercising and where at rest, okay? And the big change that you see is the shift to skeletal muscle, heart, and we should also include in here lungs, right? Notice what stays constant. The brain stays constant, right? And the other systems have a small drop, but not a tremendously small drop, right? Why do we see reduced flow to the abdomen and to the kidneys? Well, that's the result of the action of epinephrine, right? Epinephrine is going to shift the blood flow away from non-essential systems and towards essential systems, right? Look at the skin, right? More to the skin. The result is more heat loss, right? And then the heart gets more in order to maintain that flow, okay? But the brain stays constant. The velocity of blood flow changes as we go from one end of the circulatory system to the other, and it's inversely related to the cross-sectional area. Basically, it's going to be fastest in the aorta because you have the smallest cross-sectional area there. It's going to be slowest in the capillaries because the total cross-sectional area of all the capillaries is very high, okay? So that part of the container is huge, and so the pressures are very low, and that helps to promote exchange, right? And allows us to dump off nutrients and oxygen and pick up carbon dioxide and waste products because of the massive surface area that we have in the capillary beds, right? So you can see here the total area of the vascular bed shoots up when we get to the capillaries, right? And the velocity of blood flow shoots down, right? Pressures here are high, pressures here are very low, right? And that slow flow is exactly what we want. Now notice the velocity picks up a bit as capillaries dump into venules, which dump into veins and so on, right? But notice here that the pressures stay low after we release from the capillary beds. Okay, and so how do we get the blood back? Well, the respiratory and the skeletal muscle pump and the valves that are in the veins. Right? Autoregulation is the automatic change in blood flow as a result of the tissue's needs. Okay, and it's controlled by changing the diameter of arterioles that feed capillary beds. So the wider the arteriole, the more perfusion, the more nutrient and oxygen delivery, and the greater the waste product removal, and vice versa, right? Organs can regulate their own blood flow by changing the resistance in their own arterioles. And the two types of autoregulation are metabolic and myogenic, okay? Metabolic refers to the concentration of certain components in those tissues, while myogenic refers to the action of smooth muscle when it's stretched, okay? So let's look at metabolic control. How does this work? Well, basically, when the tissue is metabolically active, it's going to pump out more protons, more CO2. It's going to generate more heat. It's going to make more ADP, okay? And the result of that chemical state, it's going to have less oxygen, right? The result of those chemical states are going to be vasodilation, right? Whereas if the reverse were true, the ATP were high, the pH were high, the oxygen levels were high, and the CO2 levels were low, then what's going to happen is vasoconstriction, okay? So the effects here with vasodilation, right, is the relaxation of smooth muscle and the release of nitric oxide by endothelial cells, and that helps to keep the blood vessels open to metabolically active tissues, right? Endothelins from the endothelium are vasoconstrictors, okay? Nitric oxide and endothelins are going to balance unless the blood flow is inadequate, and the nitric oxide is going to win out, and the vessels are going to open up, and the blood's going to come on in, okay? The other way that this opens up is through the release of inflammatory vasodilators, which are a response to infection and injury in order to bring more blood to the area in order to fight a potential infection, okay? And in limited scope, this is very good, but if this were to happen all over the body, the vasodilation, the systemic vasodilation, would be so great that the blood pressure would drop to the point where you would die, okay? Myogenic controls are just the result of response to stretch of smooth muscle. When smooth muscle is stretched, it constricts. When it's not stretched, it relaxes, okay? So the way this works is that under high blood pressure, okay, we're going to stretch that smooth muscle, it's going to constrict, and so it's going to maintain a lower flow into the capillary bed, whereas when the pressures drop, it's going to dilate and maintain a more steady flow into the capillary bed, okay? So the idea here is it acts opposite what the pressure changes are, okay? So the reason for this is not to damage the bed, right? If we damage the bed, let's say we didn't have this myogenic response, then when your blood pressure would go up, what happens is the pressure of the capillaries would go up and they would pop, right? And if the blood pressure were to go down and we didn't have this response, then the delivery of blood to the capillaries would be so little that you could cause tissues to die, okay? So there we are, right? Neuronal and hormonal controls and myogenic and metabolic controls, right? So we've seen all these before, right? Maintaining our blood pressure. Long-term autoregulation takes over when short-term doesn't work, right? One example of this is angiogenesis, which is an increase in the number of blood vessels growing into a tissue and supplying an organ, okay? And that happens when metabolically active tissue mass goes up, okay? So when you get more muscle, for instance, okay? It happens in the heart when coronary vessel gets blocked and grow other vessels, or all over the body in people in high altitude areas. Okay, more blood vessels happen, more flow means more oxygen delivery. Okay, in skeletal muscle, blood flow is going to vary depending on the activity level. Okay, at rest, myogenic and neural mechanisms keep it at about a liter a minute, but during muscle activity, we have to get more blood, more oxygen and nutrients in there in order for the muscle to continue to operate, right? So during active exercise, we see a condition called hyperemia, where blood flow increases in direct proportion to the rate of the activity, right? So the stronger, the faster, the more intense you're going, the more blood flow is going to happen, right? Local controls are going to override sympathetic vasoconstriction, and muscle blood flow increases as much as 10 times, and that causes us to sustain the activity level. Now, the brain is different. The brain needs steady blood supply no matter what. At 750 mils per minute, that doesn't change even if we exercised, okay? Again, metabolic controls. How does this work? A drop in the pH is often the result of an increase in CO2 in cerebrospinal fluid, and that's going to promote vasodilation, all right? Myogenic controls, a decreased MAP is going to cause cerebral vessels to dilate, and vice versa. The brain is vulnerable under extreme systemic pressure changes. If the MAP drops under 60, we faint. That's syncope, right? If it's above 160, then we can get swelling in the brain, or we can get a stroke, okay? So we have to keep it within those areas. Now, the skin, remember, is both a blood reservoir and a thermoregulator, right? So blood is going to supply nutrients to the cells, and autoregulation is going to govern that and regulate body temperature, and that's a neural response, primarily sympathetic stimulation of the arterioles feeding the skin and the sweat glands, right? And the neurally controlled blood reservoir is important in a rest and digest situation, right? We can mobilize the blood at the periphery to serve stuff in the core, all right, when we're in rest and digest, and vice versa, right? Blood flow to venous plexuses under the skin is going to regulate our body temperature. It varies from 50 mil per minute to 2.5 liters a minute, depending on how hot we are, right? If we're super hot, we're going to be up there around 2.5 liters a minute. And if we're cold, we're going to be down around 50. And the sympathetic division of the autonomic nervous system is responsible, right? As temperatures rise, the hypothalamus signals a reduction in vasomotor stimulation of skin vessels. The blood flows from the periphery to the core to preserve heat, okay? And the blood flows from the core to the periphery to lose heat, okay? And the heat also leaves through radiation, okay? So this is what happens on a hot day, okay? On a cold day, it's the reverse, right? The skin turns pale to blue, right? And the blood gets routed from the periphery to the core, and we see an increase in piloerection, right? You get goosebumps, and the effort there is to keep your vital organs working, right? And you can suffer a little skin damage, like frostbite, if it means keeping you alive by keeping your vital organs working. Sweat also causes vasodilation as a result of bradykinins in your sweat, and that causes nitrous oxide release, okay? As the temperature drops, the blood goes to the deeper, more vital organs, okay? And remember that that's just another example of negative feedback. Now, the pulmonary circuit's a little different, okay? Pulmonary circuit is a short, low-pressure circuit between the heart and the lungs. The arterioles are more like venules, and the arteries are more like veins. The arterial resistance and the pressure are very low because the distance is very short. And the autoregulatory mechanism is opposite what you see in most other tissues, in that low oxygen causes vasoconstriction and high oxygen causes perfusion, vasodilation, right? And that allows something called ventilation perfusion coupling, in which the lungs are able to get the blood flow and the oxygen supply to meet up in the most effective way so that you oxygenate the blood more efficiently. During ventricular systole, when the ventricles are squeezing, the blood flow to the heart muscle decreases because you're mashing down on the coronary arteries. And then when the ventricles go into diastole, we perfuse the myocardium because the arteries aren't being squeezed anymore. Okay? At rest, we get a blood flow of around 250 mil per minute to the myocardium. Control is largely myogenic, and during strenuous exercise, the coronary circulation has dilation in response to vasodilator accumulation, so blood flow can go up almost 4 times what it is at rest, right? Note that cardiac cells use 65% of the oxygen, so increased blood flow means more oxygen to the tissue. Now, in capillaries, we have a situation in which we want slow flow because of the thin walls and the extensive surface area of the capillaries. Okay, the fact that you're opening and closing blood flow into these beds reflects a reduction in the speed with which the blood moves through them, and that is desirable because that means more time for exchange. Okay? Remember that the way that the gases, nutrients, and waste products move is always going to obey diffusion. So we're always going to go from high to low concentration, regardless of whether we're a dissolved gas or a nutrient or a waste product, right? So in metabolically active tissue, arterial blood is going to bring oxygen and absorb CO2, okay? And then in the lungs, the CO2 is going to flow from the blood into the lungs, while the oxygen flows from the lungs into the blood, okay? Obeying the shift in the gas concentrations, all right? How about nutrients and waste products? Well, in metabolically active tissue, nutrients flow from blood to tissue, and waste products flow in the opposite direction, okay? Lipid-soluble molecules can go wherever they want. There's no limit on what organ system they can go into. And that's one of the things that causes us to be capable of overdosing on them, and what allows them to accumulate in places like our adipose tissue. Water-soluble solutes pass through clefts and fenestrations, which are openings in the endothelial layer, while bigger molecules, like proteins and formed elements, tend to stay in the blood, okay? However, there are some molecules that are transported through penocytosis or caveoli, which are forms of vesicle-mediated active transport. So you can see here again the makeup of a continuous capillary, right? And the dynamics of the material moving through the capillaries, right? We've got paracellular and transcellular transport, right? Paracellular transport goes between the cells of the endothelium and into the surrounding tissues, and transcellular transport goes right through the cell from the lumen of the capillary into the interstitial space between the tissue cells, okay? So we have both passive and active mechanisms at work here. Fluid leaves capillaries at the arterial end and is reabsorbed at the venous end, but not all of it gets reabsorbed. Okay, some of it gets left behind, about 15%, and that ends up going into the lymphatic system, where it's going to be cleaned of things like cancer cells and pathogens at structures that are called lymph nodes. And then that cleaned fluid is going to be returned to the blood at the subclavian veins up by your collarbone. Okay, If we block that flow, then we get massive fluid in the tissues, and that causes edema. Okay. The direction and the amount of flow depend on both hydrostatic and colloid osmotic pressure. So the way that this works is that at the arterial end of the capillary bed, the major player is going to be the capillary hydrostatic pressure, okay, because it's much higher than the pressure in the tissues. The result is that you're going to push fluids into the tissue space, all right? Whereas the major action at the venial end of the capillary bed is going to be the colloid pressure, okay, that pulls the fluid back in through osmosis, all right? So you lose fluid at the arterial end, you gain fluid at the venial end, okay? So the oncotic or osmotic pressure, if you like, is created by the formed elements in the non-diffusible plasma proteins, and that draws water in, and that's the big player at the venial end of the capillary bed. Remember that interstitial osmotic pressure is really low, almost non-existent, and so that's what governs the pull. So if you look at the net filtration pressure, right, this is how we calculate it, right? Net filtration pressure is equal to the hydrostatic pressure in the capillary minus the hydrostatic pressure in the interstitial fluid. That quantity is has subtracted from it the difference between the osmotic pressure in the capillary and the osmotic pressure in the interstitial fluid. So net flow out at the arterial end and in at the venous end. So more leaves than is returned, and like we said, that 15% is what gets picked up by the lymphatic system. Okay, and you can see that diagrammed here, and you can see that extra, let's get the laser pointer, that extra 15%, okay, is going right in here. Okay, so you get most of your fluid back at the level of the capillary bed, and the remaining you get back where? At the subclavian veins. Okay, so there, what you're looking at, what's happening in the capillary bed at the arterial end, right? The major force pushing out is the hydrostatic pressure in the capillary. The major one drawing in is the osmotic pressure, but since the hydrostatic is greater than the osmotic, out goes the fluid, right? So a 10 millimeter net outward pressure, and then we flip that at the other end with the osmotic pressure in the capillary being greater than the hydrostatic pressure in the capillary, and so in comes the fluid at about 8 millimeters of mercury, and the remaining 15% of fluid is what gets picked up in the lymphatic system. Now, circulatory shock happens when you don't have adequate perfusion because of volume loss, right? The blood vessels don't fill up, the blood can't circulate, and this results in inadequate flow to meet tissue needs, and tissues begin to die. Okay? In hypovolemic shock, we're hemorrhaging, we're bleeding either internally or externally. In vascular shock, we get that release of, say, histamine all over the body. And the result is that all the capillary beds open up at the same time, and the pressures go so low that we pass out, right? And then cardiogenic shock is the result of the heart not pumping, okay? And so we don't perfuse anything. So you can see here some of the events of hypovolemic shock. And in fact, I think we have something that shows this. So let's pull that up. Okay. Let's see if we can. That. Go to this. Okay. I'm not finding it at the moment. If I find it, I'll post it. But basically what's happening in shock is that as blood flow slows down, then what we see is loss of nutrient and oxygen delivery to tissues and tissue death. And so the most metabolically active tissues die first, and the least metabolically active die last. Okay. Okay. The two main circulatory patterns in the body, as we learned in lab today, are pulmonary and systemic. So pulmonary is a short loop that runs from the heart to the lungs and back to the heart, while the systemic runs from the heart to the body and back to the heart. So the longer and higher pressure system is a systemic circulation. All right, so you can see here how that works. All right, that's why the strongest chamber of the heart is the left ventricle, because it's responsible for pushing blood out to the entire body, and so it has to overcome the greatest resistance. Okay, and that's also why the lungs are located so close to the heart, right? Because with a shorter distance, you're more effectively going to oxygenate all the blood and get it back to the heart in time to get it out to the body. Okay, so again, right, the blood flow to the lungs is from right ventricle to pulmonary trunk to pulmonary arteries. Pulmonary arteries get oxygenated blood as a result of diffusion. Pulmonary veins bring that oxygenated blood back to left atrium, from there to left ventricle, and left ventricle to aorta, and aorta to the body. All right, and that's the whole shooting match right there. Okay, where these things happen and the relative pressures, right? And remember that under normal circumstances, okay, that the amount of blood leaving the right and the left side of the heart are always the same. And if they're different, we have consequences, right? If there's more blood leaving the right side of the heart than the left side, we're going to get pleural effusion, fluid in the lungs. And if there's more blood leaving the left side of the heart than the right side, we're going to get peripheral edema, which is fluid in the extremities. And eventually, in both cases, you're going to have fluid accumulation everywhere, right? So you can see some of the differences between arteries and veins, right? There's a nice little summary chart. Where do blood vessels come from? Well, mesodermal cells, which are found in the core of something called the embryonic disc. At one point during your development, you look kind of like a frisbee with a groove down its back. Okay, I know that's hard to believe, but it happened. And that frisbee had 3 cell layers in it, right? It had an ectodermal layer, which was on the outside of the frisbee, a mesodermal layer, which was in the core of the frisbee, and then lining the hollow spaces running through the frisbee was an endodermal layer. Okay, ectoderm, mesoderm, endoderm. So mesoderm is going to form blood islands, which are going to generate vascular tubes, and chemicals are going to guide their growth. Vascular endothelial growth factor, as sometimes abbreviated VEGF, determines whether the vessel becomes an artery or a vein. And then the heart pumps blood by the 4th week of development, right? So the heart actually starts beating before it has blood to pump, which is a little unusual, but there you go. And then fetal shunts are going to be in that circulation to bypass the non-functional lungs and the non-functional liver, right? Because the pulmonary circulation while you're in your mother's womb is worthless because your lungs are filled with amniotic fluid, and your liver isn't fully developed, so there's no point in sending blood to the liver to be treated because the liver can't do any treatment, all right? So you might wonder, well, then when we're in the womb, how is it that we maintain the state of the blood? and that's through an organ called the placenta. Okay? The umbilical vein and the arteries circulate blood to and from the placenta. Congenital vascular problems are quite rare, but sometimes you have things like patent fetal structures, such as a patent ductus arteriosus or a patent foramen ovale. And if we don't surgically fix those, you can put an extra load on the heart. Okay. Vessel formation occurs in order to support the growth of the body, promote wound healing, and to rebuild vessels that are lost during things like menstruation. As you age, varicose veins, atherosclerosis, and higher blood pressure occur. Why? Well, varicose veins are the result of valves, and especially in the lower extremities, becoming incompetent and rupturing. And the result of that is the blood pools there. Okay. The increased blood pressure is the result of the stiffening of the walls of the arteries so that they don't expand as easily when blood flows through them. So the pressure in the whole system goes up. And all of these are going to contribute to hypertension as we age. Okay. This is one of the big things we worry about is the balance between hypertension and coagulation, right? So if